Your muscle is made up of many fibers called fascicles, which in turn are made up of individual muscle cells called myofibers. Myofibers are made up of myofibrils, which are essentially the contractile organelles of skeletal muscle. The sarcolemma surrounds each individual myofibril, and underlying the sarcolemma is the sarcoplasmic reticulum. In a relaxed muscle cell, the sarcoplasmic reticulum stores calcium ions. Release of calcium ions from the terminal cisterns of the sarcoplasmic reticulum triggers muscle contraction, which I will explain in greater detail in just a moment. Mitochondria, also known as the powerhouses of a cell, are prevalent because they supply ATP, which is one of the most important molecules involving muscle contraction. Along the sarcolemma are thousands of deep, tiny invaginations of the cell membrane called T-tubules, which tunnel in towards the center of the muscle fiber. Action potentials travel along the sarcolemma and through the T-tubules, spreading the signal throughout the entire muscle fiber. A myofibril is composed of both thick and thin filaments. Thick filaments are composed entirely of a protein called myosin. The myosin tails form the shaft of the thick filaments, and the myosin heads project outward toward the surrounding thin filaments. Thin filaments are made up of three molecules called actin, troponin, and tropomyosin. Actin can be recognized as a twisted chain surrounding tropomyosin. The active site on the actin molecule is covered by tropomyosin. Troponin is bonded to tropomyosin, but can also bond with calcium. Also, an extremely important part of muscle contraction. When calcium attaches to troponin, it causes tropomyosin to slide off actin, revealing the active site on actin, thus allowing the high-energy myosin heads to bind the active site on actin. The filaments inside a myofibril do not extend the entire length of a muscle cell, but are arranged in compartments called sarcomeres. Z-discs separate one sarcomere from the next. Thus, a sarcomere extends from one Z-disc to the next. The darker, middle part of the sarcomere is the A-band, and the I-band is the lighter, less dense area that contains only thin filaments. The H-zone in the center of each A-band contains thick, but not thin filaments. Let's say you go to lift up a damsel in distress. Your brain will send a message to the nerves in your arm saying, "Hey, go pick up that damsel in distress." That message is called an action potential. The action potential will travel through your body until it reaches the nerves that are involved with causing the muscles in your arm to contract. When the action potential reaches the synaptic knob of that nerve, the wave of depolarization causes calcium channels to open very wide. Calcium channels flows in, flooding the synaptic knob, which triggers synaptic vesicles containing acetylcholine, a neurotransmitter or chemical signal, to move towards the membrane. Acetylcholine is released via exocytosis in the synapse between the motor end plate of the muscle and the axon of the nerve. The acetylcholine will migrate across the synapse and bind with acetylcholine receptors on the motor end plate of the muscle. When this happens, sodium and potassium channels open, allowing sodium to flood in and potassium to flood out of the muscle. This causes a great depolarization in the muscle cell, thereby continuing the action potential initially brought by the axon.
Okay.
Calcium floods into the cell until it binds to troponin, thus causing tropomyosin to pull off the active site of actin. High-energy myosin heads leap and bind to the active site on actin, thus creating a cross bridge between the two filaments. The myosin heads use the energy, ATP, thus converting the ATP to ADP. As soon as another ATP molecule binds to the myosin head, it causes it to break free, thus repeating the process until there is no action potential. After contraction has occurred and there is no longer an action potential, leftover calcium is pumped back into the sarcoplasmic reticulum and resequestered by calsequestrin, allowing tropomyosin to once again cover the active site on the actin, which results in the relaxation of the muscle.